Hello and welcome to the NACFC live Facebook session. I'm Rebecca Schroeder. I am a CF mom first and foremost. I have a background in chemistry and I've been attending the NACFC for the last 7 years. It's really one of the most exciting weeks of the year for me. It represents the culmination of everything that we do in the community. All of the fundraising, all of the advocacy work that we do is really to pave the way for the work that we talk about, the research that we talk about here at this conference. So I'm very excited to be joined today by two gentlemen. We have Dr. Christopher Goss, who is the Professor of Pediatrics and Medicine at the University of Washington Medical Center, as well as the co-executive director of the Cystic Fibrosis Therapeutics Development Network, which facilitates and develops clinical trials so important to our community. I'm also pleased to be joined by Dr. Patrick Flume, who is a professor of medicine and pediatrics at the Medical University of South Carolina, as well as center director at MUSC. Both of these gentlemen are here at the conference for the umpteenth time, I assume, to present their work. Now, I know from the family perspective, the exacerbations and infections in cystic fibrosis can be very burdensome and worrisome for families and individuals to deal with. You're both working on some really exciting new research. Dr. Goss, I know you have in your work some new non-antibiotic approaches, some novel approaches to treatment. Why don't you let us know a little about that?
It is an exciting time for CF, and I think the CF community has reached out with support from the CF Foundation to try to develop really novel approaches to treating infection, which is so central to CF. And there are two, I think, very innovative approaches. One is a study that I lead with Dr. Pradeep Singh, and we're using intravenous gallium nitrate. And gallium is a metal, but it turns out gallium is a very interesting molecule. It's very similar in size to iron, and bacteria need iron. That's our main food source. And it's been shown really nicely that giving gallium to Pseudomonas grown in an agar prevents it from making biofilm, or this coating around it that protects it from your host, from you, and also kills Pseudomonas. And it can also kill a number of other bacteria that have been very problematic in CF. So it's a very novel approach. We think it gets in and poisons the bacteria. And so we're in the midst of a very large clinical trial. It's a phase 2 proof of principle trial called the IGNITE study. And there are 23 centers around the United States that are participating. And enrollment has gone great. We're actually probably going to be finishing up in January with the last patient, last visit, and hopefully we'll have a data lock and then results late in the spring of 2018. And we hope to be presenting those results here. But we think this is a novel approach because it basically takes a metal and tries to kill bacteria, and even bacteria that are highly resistant to the antibiotics we are currently using to treat CF.
Huge problem in cystic fibrosis. Incredible that you're finding these new pathways. You have another study that you're looking at as well.
And I should note that actually Dr. Flume and I are PIs on all of these studies together. The next one is a study, a use of inhaled nitric oxide. This is a gas, and with the support of a CF Foundation, a company called Novateris is doing a phase 2 trial on inhaled nitric oxide to treat chronic pseudomonal infection in the lung of CF patients. And nitric oxides are interesting, and it has a similar property. It causes dispersion of Pseudomonas biofilm. It doesn't allow them to make biofilms, and actually can kill Pseudomonas and fungi and other nasty bacteria like non-tuberculous mycobacterium. And this is a very intensive study. Patients are in the hospital daily for 8 days. It's a very innovative approach to think about a gas killing bacteria in the CF lung. And so hopefully the study is already enrolling, and it certainly represents a very different approach to treating a chronic infection.
Absolutely. Now in the community, we call that biofilm the super slime. So breaking down the super slime with some of these new techniques. Now, Dr. Flume, I know you've been involved in these studies as well, but you're taking a little deeper look at exacerbations also. Let's talk a little bit about that.
Yes, so as you know, pulmonary exacerbations is a very common complication in our patients. Several years ago, I was co-chairing a committee, Chris actually was on that committee as well, where we were looking at defining the best practices for pulmonary exacerbations. And when we searched the literature, we found that there wasn't a lot there to tell us what really was the best practice for simple questions like how do I choose antibiotics, where should I treat patients, how long should I treat them. So the CF Foundation asked Chris and I to co-lead a group to try to define what would be best practices. And this led to our program that we call STOP, for Standardized Treatment of Pulmonary Exacerbations. Mm-hmm. After our first observational study, we've now launched what we call STOP 2, which is the first interventional study. It's a very large study. We're going to enroll a little over 1,000 patients. Wow. To test durations of IV antibiotics, under the premise that there's such a thing as treating for too short a period of time where a person doesn't get all the way better, but also there's risk of treating for too long, where there's really no added benefit, but just added cost and potential toxicity. So we're moving right along. We're actually ahead of schedule, so we were very excited about that.
So this will allow us to really refine our treatment of an exacerbation and really choose the best duration, the best practices to eradicate that with the fewest side effects.
And we think it's really just the first step. So you identify the right duration, which then allows you to again look at other questions, which could be where's the best place to treat? Should you use steroids? How do you choose antibiotics? So the goal is that we build these studies that eventually have a long list of best practices.
Right. So we discussed some of these novel approaches that we're looking at to treat infections. I know that you are also working on a new way to deliver an already used antibiotic. Tell me about that study, Dr. Flume.
So we just launched a study to treat patients who have MRSA. Essentially, this is repurposing an antibiotic, in this case vancomycin, by the aerosol route. And this is very similar to what has been done for Pseudomonas. So we feel very comfortable about how to treat Pseudomonas, whether it's an eradication approach for first infection or chronic suppression of people who have chronic infection. And so we've learned all that for Pseudomonas, but then people can ask, well, what about staph? What about Stenotrophomonas? Should we use a similar approach? MRSA has become a much more prominent bug in our patients. We think those patients are sicker, and so basically we're using that same strategy. And so we just launched our phase 3 study looking at a inhaled formulation of vancomycin for the suppression of infection.
Right. So what are some of the advantages of inhaling an antibiotic versus taking it intravenously or orally, for example?
Right. So some medications are not available orally. So for example, tobramycin is not available that way. Vancomycin isn't absorbed, so we don't use it to treat systemic infections. So in some cases, it's either intravenously or inhaled.
I see.
One of the advantages of going an inhaled route is you can get very high concentrations at the site of infection, which for some types of bugs and drugs, that's the best way to kill them, and yet have low systemic toxicity. And so when you're talking about using a medicine for a chronic treatment, like we would with aerosol, most patients are not going to want an IV for long periods of time, so the aerosol route becomes an attractive method.
Well, absolutely. You know, in terms of the burden on patients and families, the aerosol sounds like a very attractive option coming down the pipeline. So I'm very excited to hear that we're developing new ways, not just new antibiotics and new treatments, but new ways to take old ones as well. So thank you both for the great explanations of your work. And we would like to take the time now to turn to our live audience they all have some great questions for you to field today. The first one is from Michelle, and she asks, what about using inhaled colloidal silver to kill bacteria in the lungs? I've read a little bit anecdotally about this. What's your opinion about that, doctors?
It is a great question. It's not an uncommon question, particularly to our group because we are studying another metal.
Right.
Colloidal silver is an antimicrobial. It's been used in wound creams called sulfadiazine. It's been used coating catheters to prevent infection on catheters. The challenge with moving forward with a nebulized silver is that it would need the same structured approach that we've done with gallium to ensure safety of inhalation. There is an entity of silver toxicity.
Right.
You can actually have nerve damage, and your skin can turn an interesting color of gray. So again, I think understanding the risks and benefits, we don't recommend any of our patients do that. The development of an inhalational agent is very challenging and requires very careful oversight by the FDA. So we don't recommend our patients do that.
So there just hasn't been the process of clinical trials and assurance of safety that other drugs have gone through that rigorous testing. So we're going to cite on the area of safety and take the next question here. So what happens if you're allergic to vancomycin? Could you take an antihistamine or something to mitigate that response before treatment and still be eligible for that type of treatment?
So the challenge there is separating out what is a true allergy from what is just an untoward reaction, intolerability.
I see.
So if someone is truly allergic, then they should not be taking a medicine like that, and we would not be recommending even an antihistamine to try to prevent that. But sometimes there are reactions that aren't allergic, and with vancomycin, the most common one is red man syndrome, which when you give it intravenously, it does release histamine and cause the red man syndrome. And you can combat that by actually slowing the infusion. We think our patients can do okay with the inhaled route and not experience that kind of an adverse event. So that's not an allergy, but true allergy, don't do it.
So true allergy would be no inhaled, no intravenous.
Correct.
If you're just having this reaction response, then there's a potential there that you may lessen it with the inhaled version. Great. So we're going to take another question. Bacteriophage research and development therapy, is that an option that you see as an alternative for antibiotics in the future? I've seen some talks about this, and it's a very exciting concept. What about therapeutically?
So I'll start, but maybe Patrick can also comment. Bacteriophage therapy, for those in the audience that maybe haven't heard about it, is to use what are really viral particles that actually kill bacteria. Bacteria, like humans have pathogens, bacteria have pathogens, and the feeling is that bacteriophages are the pathogens of bacteria. And it makes sense that you could use this pathogen to kill bacteria. It has had a lot of challenges in development, and one of the most challenging things is creating a unified batch where it's always the same, which is how drug development has to move forward. And that's probably been the most challenging aspect of its drug development. There are certainly places in the world you can get bacteriophages, and I've had very mixed results from that, patients going abroad to get them, including severe illness.
Okay.
So outside of the regulated drug market, I think patients are potentially at significant risk of either fraud or danger from a toxic inhalation or toxic therapy. So
Okay.
I think they have a lot of challenges to moving forward with bacteriophages that particularly when you think about developing a drug, I don't see an easy solution, but I'd love to hear Patrick's thoughts.
Well, probably more than I should say here, but one of the challenges in terms of how I understand the biology is the bacteriophages don't kill bugs dead. So they
Okay.
kill them, but they also need them to be a host to continue to, reproduce. And so it could be looked at, well, that might still be okay if you're just trying to get a static effect where you're just suppressing it. You don't need to kill everything completely. But really more, but is what Chris has said, you have to have a consistent, stable product that you're using. So when you use the product today, it's the same as the product you use in the future. And then in terms of developing resistant patterns, that's a whole nother session.
Well, I know that safety is obviously concern to all CF patients and families. We don't want to do anything, obviously, to further burden the health of an individual living with cystic fibrosis. So standardizing those treatments, probably the best way to go, and wait for that result at maybe the future NACFC meeting, right? So I just wanted to thank you both personally for the work that you do, the time and expertise that you dedicate to individuals and families living with cystic fibrosis. It means the world to us. Thank you for taking the time to travel here to present your research and to share all of the exciting news with the community and our Facebook Live crowd at home. So thanks for joining us. You can continue watching NACFC live feeds from the NACFC for the next couple of days, and we'll see you soon.